So the beginning of tissues now, and you know the four tissue types, you know the four basic tissue types. Of course, this matters because tissues are groups of similar functioning cells. So we have epithelial, and then we have muscle tissue. Maybe just mute the mics if you can, and just so I don't have to stop. And then you have connective tissue and nerve tissue. So now in lab, you've seen all this before. You know all the different types of epithelial tissue, the simple, the stratified, the pseudostratified, the squamous, the columnar, the cuboidal, and mixing in the stratified and simple and pseudostratified. So for the lab practical and for the lecture exam, you should know what it looks like, mostly for the lab practical, identifying it, and then you have to know the function of it, and then you have to know the places where you might find it. So the different types of epithelial tissue. And muscle we're going to do in great detail, like I keep saying, but you have to know the difference between skeletal, cardiac, and smooth muscle, also known as visceral. This is connective tissue is probably the most difficult one. You know, you have the proper dense connective tissue, proper, loose and dense, and then the specialized. And the specialized is bone, cartilage, adipose I would throw in there, but even though that's loose, that's kind of not really specialized. Adipose, but that's an easy one to see. Adipose and blood. So you should be able to identify all of those, everything. And nerve is just one thing. It's just show you can look at the nerve, the neuron, and its parts, like the cell body and the cell body nucleus, the axon, which is that wiry fiber thing, looks like a kite. And then you have the glial cell. And mostly that's the nucleus of the glial cell. Remember, nucleus is always dark staining. So this is a nice review for lab. You know, as we're going through this, and I'll tell you what's important for the lecture too. So tissues are a group of similar structure and function cells, anatomy and physiology, very specialized for this tissue. Histology is the study of tissue, and that's what we did in lab. And tomorrow we'll have actual slides to look at, unless we get a tsunami or an earthquake or snowstorm. Tomorrow we'll be in lab and we'll be able to do it, and we'll look at everything. And we'll look at the skin too. So this is a good double up lab. This one's pretty good, where we can manage it very easily. Like I know I spent a lot of time last Thursday, but that was good too. And you have that recording. So you guys, you got to do good. You got to do good. And we'll do good on the final. It's all cumulative for that final. I don't give you a cumulative exam, but you get a nice summary of everything that you've done in one like multiple choice and ordering test. So that's really nice. So nervous tissue, the brain, the spinal cord. Like if you see a neuron, where do you find that? I'll tell you where I find it. I find it in the brain or the spinal cord. Nerves I don't love, but you could say I find it in the spinal nerves. That would be really good. But think brain and spinal cord, because that's the central nervous system. Muscles should be easy. You just kind of say, where do you find them? Like you could say the heart, like if it's cardiac muscle. And what is the function? They contract, and the heart moves blood, pumps blood, and the skeletal muscles move joints. Yeah. Epithelial tissue is about covering and lining things and secreting, sometimes absorbing. Remember, glands are epithelial tissue. What they secrete, which is hormones, which is endocrine. Endocrine glands secrete hormones into the blood. Endocrine means into the blood. Then you have exocrine glands, which secrete like enzymes, or like salivary enzymes or digestive enzymes into ducts, or sweat glands secrete sweat into the outer portion of the body, or cerumen glands. Like, remember, we talked about the ear glands in the outer ear, the ceruminous glands that secrete cerumen. Those are all exocrine, and they're epithelial tissue. The sebaceous gland in the dermis that secretes sebum into the, or out of the skin, actually. Connective tissue is a little more complicated, but it depends on which one we're talking about. So you could say support and protection. Sometimes you could say epithelial tissue protects too. And bone tissue, which is connective tissue, obviously protects, connects with tendons. Fat is insulation. It depends on which connective tissue. So that's the more complicated one. And we're going to cover bone extensively, starting maybe tonight or next time. So microscopy, which we're doing in lab, something you can't see with the unaided eye. Okay, so this is more lab. We use light microscopes, of course. We don't have electron microscopes at CCM. Light compound microscopes. And remember the different objective lenses and the magnification of the ocular lenses. So don't forget all that stuff for the practical. And staining, we didn't really do that, but we could have if you tried to get a little scraping out of your cheek. There's methylene blue or iodine to stain things. And if you look at the tissue that we're going to see, like this is histology on this side. It has stain, so you stain it with things like iodine or biuret, a Benedict solution, all these things that we use so we can highlight specific structures like cells and adipose tissue or microvilli or cilia. This is a real picture though. We're not going to see too much of that. So epithelium, two main forms, the kind that covers and lines, and then there's a secretory, glandular. All right, so know the difference between, I'm going to ask you this on the exam, the difference between endocrine and exocrine. Endocrine, the glands secrete their products directly into the blood. That's why endo, into, within. Exocrine is like a salivary gland, which secretes things into a duct or a tube or out of the body. Functions, here they are. This is really going to be helpful. Protection, right? Epidermis protects the stratified squamous epithelium with keratin in the epidermis and the non-keratinized stratified squamous epithelium in the esophagus or the vagina. Then we have absorption. Remember absorption, that simple columnar in the intestines, because we're absorbing the nutrients. Remember that ridiculous chemistry lecture? So you're here all night talking about how carbohydrates are broken down into monosaccharides that need to be absorbed like glucose, or proteins are broken down through hydrolysis into amino acids. So remember, building up like monosaccharide plus monosaccharide to a disaccharide, that's dehydration synthesis, because we're building up something bigger. And that's dehydration, means you lose the water in that. Hydrolysis is the opposite. If we take sucrose, which is a disaccharide, we break it down, that's hydrolysis, which we add a water. Know those two reactions. So we have to absorb glucose, amino acids, and fatty acids are the breakdown product of triglycerides, which are lipids. So know all those macromolecules best you can. You're going to do great. Filtration, one of my favorites, right? Simple squamous epithelium is simple squamous for diffusion too. Diffusion and filtration. Excretion, like the kidney tubules, sometimes they have to excrete things, so they'll secrete or reabsorb. Secretion, mostly those simple cuboidal cells, also in the kidney. All right? And sensory reception is in our skin, but that's really nerve tissue. That's really nerve tissue, but it's in our skin. So remember, this is avascular. Epithelial tissue. We're still talking about epithelial tissue, but innervated means it has nerve input. That's why I just mentioned that on the slide before. Polarity, you remember the cell membrane? It creates a polarity, positive on the outside, negative on the inside. The phospholipid membrane, right? The phospholipid membrane, the phosphates, it's a biphospholipid membrane. So the phosphates are polar, the balloons. The tails are all lipid, non-polar, hydrophobic. So get those terms down for the exam and see how it relates to tissues. So lipids are hydrophobic, fear water, right? Something that, and they're nonpolar. Lipids are nonpolar. Like a phosphate in this case is hydrophilic, and it's polar, like water. So it's hydrophilic. That's why it loves water. It's because it's polar. So get this. The three macromolecules we're talking about, proteins, carbohydrates, and lipids. Lipids are nonpolar, hydrophobic. Carbohydrates and protein are very polar, very hydrophilic. Get it straight. Regeneration, and this is where I talked about in lab last week, is where you undergo a lot of mitosis. So they're very mitotic cells of the epithelial tissue. Mitotic means they divide. The nucleus divides to make exactly the same cells over and over. And this goes on and on, especially in the epidermis of the skin. So here's just going through some of the parts. Remember, the apical surface is the top, and that's where you have things like cilia and microvilli on the top of the cell facing the lumen. So you have to know what a lumen is. A lumen, right? If this is a tube, right, and it's lined with, say, simple cuboidal epithelium, you get it, right, if I keep going around. The inside of the tube is the lumen. That's where either the filtrate of urine is going through, or blood goes through a lumen in the blood vessels. The breakdown products of Popeyes goes through the small and large intestines, lumen. The esophagus has a lumen. The trachea, which is for oxygen, carbon dioxide, and other gases to go into your lungs, has a lumen. And the trachea is surrounded by pseudostratified columnar ciliated epithelium. This is the way you got to start thinking now for this part. And the bottom surface is the basal surface. And there's a basal lamina, which is really, we like to call the basement membrane. You just have to know that. You don't have to know the two layers. Basement membrane. So all epithelial tissue is attached to that basically connective tissue basement membrane. So the cells are all like a simple squamous epithelium, like in a blood vessel, is all one basement membrane, one layer of cells, simple. And then if we pile them up, then it would be stratified. And there's reasons for that. Is it for protection? Yeah, if it's squamous, it's for protection. Simple squamous epithelium, just one layer, is more for filtration and diffusion. You're going to see that over and over again. So again, here's a lumen up here. A lumen is all out here, all in here. So here's you can see the nuclei. Do you see that? You see that. I know you see that because it's nice and darkly staining. You can hardly see a basement membrane, but it's usually there. And you can see that this is simple columnar, right? If it had cilia on it or microvilli, then that would be microvilli. So columnar could have either cilia or microvilli, depending on where it is and what it needs to do. So this is the connections between cells, like what holds them together. So the desmosomes are little proteins that connect the epithelial tissue to the basement membrane. Tight junctions are usually between the cells. So tight junctions we need, we don't want leakage. Desmosomes were allowing for diffusion, filtration, and other movement. Remember, epithelial tissue is avascular, so it has to really rely on that diffusion from the extracellular fluid. So tight junctions are important, like in the blood-brain barrier, in the bladder, the uterus. Things that you want to keep the contents in the stomach too. We don't want to lose that acidic part of the stomach, right? So supportive connective tissue, and there's a reticular lamina, but really what I need you to know is the basement membrane. But it's the two lamina, basal and reticular lamina. Reticular is always protein, like you saw in connective tissue, right? Reticular connective tissue that gives support and structure. Yeah. So that reinforces the epithelial tissue. And this is important. I know that we don't talk a lot about it, but this is important for holding your tissue together, because if they weren't here, those desmosomes and hemidesmosomes and other connective adherens connections, we would have ripping apart constantly of the tissue, the epithelial tissue especially. Yeah. So cancerous cells, this is kind of bringing up a clinical response. Cancerous cells are cells that undergo crazy mitosis. So those cells kind of have a mind of their own and a destination of their own. So they kind of do not stay contained and they grow out of control. Like, remember in lab we looked at like a melanoma, which is cancer, or even squamous cell carcinoma and basal carcinoma. They're cancers of the skin, epithelial tissue. And you could see how we looked at the A, B, C, D, E rule, and you could see how they just spread out within that tissue. But that could happen in any tissue. It could happen in the lungs. It could happen in the pancreas, the liver, the stomach, the kidneys, anywhere that there's epithelial tissue. Because you should know this, epithelial tissue of the 4 tissue types is the most prone to cancer. It's true, okay? So they can invade other tissues and spread. Spreading of cancer is called metastasis. Metastasis. So if it's in one place, it's called in situ, and it's controlled. Like a basal cell carcinoma is usually in situ, in the same place. Metastatic, like especially something like melanoma, which gets into the blood and neighboring tissues, is very malignant. I mean, any cancer that's mitosing abnormally is malignant. It's not benign. Benign is not cancer. But one is spreading faster than the other, and that's epithelial tissue. So no blood vessels, avascular. Whenever you see an A in front of something, that means that it's not there. Avascular. So they need this diffusion. They need that. Remember all those transports I dragged you through? Remember, passive transport is always free. Diffusion is passive transport. Osmosis is diffusion of water. Therefore, it's passive. Doesn't need ATP. Active transport is the one that is costing you ATP. So when you go from a high concentration to a low concentration through a membrane, that is diffusion. When you go against the concentration gradient, going from low to high, that you're going to need ATP. Therefore, that's going to be considered active transport. Cool. So we have innervation, which means nerve fibers are supplying the epithelial tissue, because you have sensory nerves in your skin, your epidermis, but you also have to have the nervous system control the output of glands, because glands are effectors. Remember this, it seems like so long ago, but effectors, or what does the work? Like, remember the feedback mechanisms. Remember when you're shivering, the skeletal muscle is the effector when you're cold. When your body temperature goes up in a hot day, exercising the heat, and your sweat glands become the effector to release water. So know what an effector is, know what a stimulus is, know what a control center is, and a receptor. So it starts out, what, receptor? And usually there's a stimulus to the receptor. Then there's an input to the control center. Then there's an output from the control center to the effector. And that's going to change the condition. But you don't want to overshoot the condition, so the output has to stop. Therefore, it's negative feedback. Positive feedback is the opposite. So for the exam, make sure you know the difference between positive and negative feedback. Chapter 1, right? Seems like years ago. Regeneration, highly regenerative. Yeah. So when the cells start to lose their polarity, they begin to undergo mitosis. Or if there's a problem like friction, because they're for protection, or hostile substances like acids. So they're replaced and they need to undergo cell division, nuclear cell division, which is mitosis. Okay, and this should all sound familiar, and this is a great review for both exam 2 and, of course, the lab practical opening on Sunday. So you have either simple epithelium or stratified epithelium. Simple is, remember, just one cell layer connected to the basement membrane. Okay, stratified is one layer but then there's layers on top of it. So even if there's 2 layers, it's still stratified. And then the shapes, the squamous from the lateral view, the cuboidal, usually central nucleus, and the columnar, which usually has this nucleus down here. So be able to identify the membrane, the nucleus, and the cytoplasm, which is anything between the nucleus and the cell membrane. So they're named according to their top layer for the shape. Like these are paver stones looking, so these are going to be a squamous, right? But in this one, same thing. These are still squamous, but this one's stratified, right? The bottom one's stratified, and the top one's simple. And the apical surface is all the top, and you can see they look like paver stones in this case. Yeah, of course, it's hard to see the cuboidal versus columnar, but there'll be no tricks. So this is the shapes. You can see the little pink nuclei in there. So simple epithelial, like simple squamous epithelium. You should know what it looks like, right? Where when you see it. Sometimes there's a lumen, like don't forget this was important in the air sacs of the lungs, the alveoli, right? So here we want diffusion and filtration. So diffusion is really important for the carbon dioxide and oxygen, O2 and CO2 in the lungs, diffusing back and forth. We want to get carbon dioxide out and oxygen into the body and into the blood. Kidneys are more about filtration. The glomerulus of the kidneys will filter our blood. The kidneys, that's the job it has. It filters the blood, then it's either going to excrete through the urine or it's going to reabsorb it back into the blood. That's why the kidney tubules, which are simple cuboidal epithelium, are mostly about secretion, right? Secretion. So, but if you said absorption, I would accept it. No problem. No problem at all. So there's different types of, 2 different types of simple squamous epithelium. So endothelium, you should remember this, lines blood vessels and lines the inner part of the chambers of the heart, and lymphatic vessels, which are kind of like veins. Lymphatic vessels are for reabsorbing excess fluid. They carry white blood cells and dietary lipids. Blood vessels are arteries, veins, arterioles, venules, and capillaries. So a capillary, like if you look at blood vessels, you should know the aorta, because aorta is the one with the elastic connective tissue. Not elastic cartilage. The aorta is a big, thick, elastic wall. Here's the lumen here. So it has 3 big layers. It's got a muscle layer. Well, first it has the endothelial layer, simple squamous, on both sides, inner and out. It's lining the lumen of the aorta, let's say. But the aorta is very elastic. So that's the one I'm going to use, which I probably did on a quiz or definitely on a practical, where you're going to see elastic connective tissue, not elastic cartilage in someplace like the aorta. So the arteries, like an artery, is, well, here's the lumen, right? Here's the lumen. And the walls are very muscular. There's muscle cells in the walls. But the inner layer is endothelium, simple squamous. Sorry if I'm not drawing that right. And don't forget that's on both sides. So you have one layer of endothelium, then you have some muscle, then you have more connective tissue. So it's got thick walls, very muscular walls, but the inner lining is still endothelium. Now, all a capillary has is one basement membrane on both sides, of course, and one layer of endothelium. So this is really where the diffusion happens in capillaries, especially in the lungs, which there's the lumen. Don't forget this nucleus that stains. So in the lung sacs, the alveoli are here, right? So this is kind of like what an alveolar sac looks like. Each one of these is alveoli. So the air comes in from the lungs. Of course, this is not drawn to scale. This will wind up in the Guggenheim, though, I guarantee it. So the oxygen comes down into the lungs. So the alveoli is basically lined with epithelium, simple squamous epithelium. So the O2, the oxygen, can just come out into the interstitial space and then get into the blood by diffusion. Of course, when you inhale, you're bringing in all the oxygen. When you exhale, and this happens on both sides, when you exhale, carbon dioxide has to get out of the blood vessel, into the interstitial space, into the simple squamous epithelium, and get the hell out of the body. We want to get this stuff out. We want this stuff into the blood and back to the tissue, especially epithelial, because they rely on diffusion. So mesothelium is more about between cavities, right between cavities. So if you have, like, here's the trachea, then the trachea, this is air coming in, just like I showed you, but now this is bigger. This alveoli are microscopic. So then you go into the lungs, right? Here's the lungs on both sides. The right side, you have 3 lobes. Left side, you just have 2. So, you know the thoracic cage, like where the ribs are out here? That's out here, right? So this thoracic cage is lined with mesothelium. So this is mesothelium out here. They call that the parietal pleura, if you remember that from lab, parietal pleura. And this happens with the heart too, by the way. So there's a cavity between the organs and the cages, or could be cages, it could just be the outer lining of the abdominal contents. But there's always space between. You want to reduce friction because the lungs are going to expand. So the inner parts are lined with endothelium. The outer parts are lined with mesothelium. Okay? So another difference, basically, right? So here we go. This is great for lab here because it's showing the description, the cartoon, what it looks like in the real histology, like, you know, in your book or in these slides, and what you're going to see tomorrow in lab, which you didn't get to see last week because of the major snowstorm. So this is all O2 you want to get in here, carbon dioxide. You got oxygen in here, so carbon dioxide you want to get out of the body. Oxygen you want to get into the blood, right? So it diffuses through here. So this is simple squamous epithelium of the alveoli in the lungs, but you just have to say the lungs. Okay, and where's the function? Diffusion, or in the kidney, glomerulus of the kidney, not the tubules. So filtration, let's just give you this word now, glomerulus, is capillaries, which are simple squamous epithelium, basement membrane, in the kidney that filters. Diffusion happens here in the lungs. Carbon dioxide and oxygen. By the way, carbon dioxide and oxygen are very lipid soluble, so they'll move in and out of membranes very easily, as opposed to something that's non-lipid soluble, like a protein or carbohydrate. They have a harder time getting through a mostly lipid membrane. Remember the membrane now, before you take the test, is a biphospholipid layer with fluid mosaic, the proteins that are all embedded in there, really important. And the difference between hydrophilic and hydrophobic. Crazy, right? So here's simple cuboidal, one layer of cuboidal cells on the basement membrane. And this you see in the kidney tubules, not the glomerulus, and ducts of glands. Ducts of glands, not the actual glands, but the ducts that they're secreting into. And the kidney has ducts too. So you didn't get there yet, so just keep it as simple as you can. So these are more secretion and absorption. So I like to say more secretion, though, for this, because that makes it easier for you to remember what the simple columnar does, which is more absorption. But they also secrete. So any epithelial tissue, there's very few that don't do both. So this is the simple cuboidal cartoon. Here's the real histology of the kidney tubules. Remember, this is the lumen right here. This is the basement membrane. Here's the nuclei of the one single layer of cells. This is a perfect example right here. So hopefully we'll get a nice pointer. What type of cell is that? And you're going to say, that's a simple cuboidal. What's the function? Secretion or absorption. Where do you find it? In the kidneys. That's all you have to say, even though this is kidney tubules. So keep it as simple. Make your little flashcards. It's a tough class. I understand. Simple columnar epithelium. One layer of columnar-shaped cells attached to a basement membrane. Tall, tightly packed. Sometimes you're looking even at simple columnar, it kind of looks like pseudostratified because it really is. Just pseudostratified has its own look. So some have the apical surface has microvilli. Remember, microvilli is for absorption. The cilia is for movement, like mucus, moving mucus through a lumen. You getting this lumen thing? That's really important. You know, lumen, basement membrane, all really important epithelial tissue, accessory things. And then you got these. Remember what these look like. So the goblet cells produce that mucin or mucin that becomes mucus in the lumens. Important. So this is a lot of absorption and secretion in simple columnar. So you're absorbing, like in the small intestine, you're absorbing the nutrients, right, from Popeyes, Chick-fil-A, wherever we are, right? I think we all got to go. I think I'm going to go on a plant-based diet and see if we can find a different place to eat. I'm reading all this stuff. So secretion is the goblet cells within that epithelial tissue. Of course, there's enzymes. There's so many reactions. They need a catalyst. They need to lower the energy of activation to get these things done. Or else it would take forever to diffuse things into, or actively transport, I should say, things into and out of cells. So ciliated cells, the cilia moves the mucus. I think you're getting this. We're getting this. Repetition is really good. So this, in the digestive tract, it's absorbing the amino acids. So here we would have microvilli. Makes sense to me, right? So gallbladder is really not absorbing anything. That's secreting bile. The ducts are surrounded of glands. The bronchi are those, like, remember I said the trachea comes down and then it branches? Well, the bronchi is this branch, and that's going to branch even further. So bronchi are down here, big tubes. So they're lined with simple columnar epithelium, if you want to use that. So you can't just say the lungs, but if you did, that would be fine. I would get it. And the uterine tubes are simple columnar, and they usually have cilia on them because they got to move that little oocyte. Remember on a Friday night, you know, where the oocyte is moved to the right position so the sperm can swim up and fertilize it, entering the nucleus, forming one cell. So what do we have here? Simple columnar. So here's the cartoon, basement membrane, everything's shown nicely if we drew it. But now we look at the real histology. That's what we're looking at a microscope. This is beautiful. Beautiful. Where's the lumen? This is the lumen. I like it. So let's see, can I find, I don't really see a basement membrane, but there's kind of a basement membrane here. And here's the nuclei of these simple columnar cells. Here's the cytoplasm. And you see this? Look really close. Right around the lumen. All these are little hairs in here. That's cilia. It's silly, I'm telling you. And look at this big thing, big pimp cup, spitting out this gooey stuff. This is a goblet cell. It's gobbing up all the mucus. So you get a cough, you could, you know, take your mask off for a second, get away from people and spit it out. Or swallow it, whatever you need to do. So in this case, this is not the respiratory system though. So I'm talking about spitting out mucus. This is already down where it's going to be digested into the fecal material, because this is the intestinal tract where you have simple columnar epithelium. So I'm just talking about goblet cells with mucus. Later on, you're going to see goblet cells in the trachea, upper respiratory, with your pseudostratified ciliated columnar epithelium. Crazy. Pseudostratified. So it looks like it's stratified, but we're pulling the wool over your eyes because it's not stratified. Why do they got to do this to us this early in the semester? But it means false. So many are ciliated, silly, right? And they have goblet cells in between. So this is mostly in the upper respiratory tract, I think, is our best bet for pseudostratified columnar epithelium, like the trachea, which technically is both upper and lower respiratory system, but Don't tell anybody I told you that. So here it is, my friends. This is it. This is the cartoon. You can see, you really can't see, but you have to take my word that these cells are all connected to the basement membrane. This is not two layered. Here's the trachea. This drawing, nowhere near as good as my drawings have been. I don't care. I don't care what anybody says. So the trachea has a lumen. And I think right now, you know, but now this is a great picture of cilia. It's silly. And here's the basement membrane. Try to find that. That's hard. And then you see the goblet cells. Let me tell you something. I'll tell you, give you a little hint. I just realized. Whenever you see goblet cells, it's probably going to be either pseudo-stratified or simple columnar. So pseudo-stratified columnar or simple columnar. Think about it. I just blew your mind. And this is the cilia. Now, the large intestine could have microvilli. So another difference for a lecture, like what they do physiologically. So stratified is two more layers. There's only one layer, the basal layer on the basement membrane, and they pile up on top. So we name the shape based on what the apical surface looks like. Yeah. So if that's free surface. So now they're changing the vernacular on us, but this is apical. Apical surface. And that's the part that faces the lumen. Faces the lumen. That sounds like a good TV show on Netflix. Face the lumen. Am I cilia or am I microvilli? But this is stratified we're talking about. So this has an area that protects wear and tear, like the skin. All right. I don't know if we really wear and tear our skin, but it's possible. Right. And the skin is keratinized. That's a protein, waxy protein that kind of waterproofs and strengthens the skin. So the skin, which is only in that one layer called the epidermis. Epidermis. That's epithelial tissue, my friends. Non-keratinized would be in a place like the esophagus. That's that tube that takes the food from your mouth, the Popeyes, from the mouth down into the stomach. Chick-fil-A tomorrow. Okay, so that's non-keratinized. You don't want keratin in there. And the vagina, too, is non-keratinized, stratified, squamous in this case. So what does it look like? What is it for? Where do I find it? This is the questions I ask myself all night. Here's the cartoon version of that. So the bottom layer could look a little cuboidal, but it's really named after the top. Yeah. All right, so this is non-keratinized stratified squamous epithelium. Here's the lumen. Oral cavity, really, in the pharynx and the esophagus. Like here's the esophagus here. Actually, the pharynx is everything in the back of the mouth and nose. So where the air and food mix down here, then the esophagus is posterior to the trachea. Let me show you if I can show you that close up. You got to know this stuff. This is anatomy and physiology. This isn't, Chick-fil-A 101 here. We got to get down to business. So the pharynx is all here. So the food and the air mix, because the air is coming in here. Here comes the oxygen, right? And it goes back through this meatus. Is going to learn all this crap in here, believe it or not. So this is lined by pseudostratified columnar, actually, in this area. But now when we get into the esophagus with the food, so the food comes in the mouth. Here comes the, let's get Popeye's chicken bone comes in there. And now the food has to go posterior down the esophagus. So the epithelium that lines the esophagus is non-keratinized, stratified squamous. Just to see if you guys are awake. You're with me? Anybody with me? Any Anthony's out there with me?
We're here.
They're all here. But that's Jeremiah, I think. Was that Anthony Granados?
Anthony.
I'm here.
Okay. So let me ask you this, Smarty Pants. So look at this. Look at what's happening. Now the food comes down here. What's this elastic cartilage? I said elastic cartilage. I didn't say elastic connective tissue. What's that flap called? that's made of elastic cartilage. There's only 3 places where we have elastic cartilage. I got to make sure you're awake. So this is covering this opening to the trachea because you don't want the food going down there. So the opening of the trachea is called the glottis, and you have your larynx, which is your voice box. So what's that elastic cartilage flap called? You could do this. Come on. I wish I had your notes on that.
Epiglottis.
Epiglottis, because epiglottis means top. This obnoxious writing and this power. Epiglottis is elastic cartilage, not just elastic connective tissue. So there are only 2 other places where we see elastic cartilage. One of them is the epiglottis. The other 2 are the eustachian tube and around your ears, like this right here, is elastic cartilage. That little satellite dish that brings in the sound. 1,000 Zoom calls a week with your earbuds in. So we have stratified cuboidal epithelium. It's rare, but it's not as rare as this one. So it's quite rare. Use quite on a practical. I'll give you an extra point. You say quite. Just spell it right. So this is glands, really, the mammary glands of the breast that are releasing milk, and the sweat glands. That's not that rare, but it's rare enough. You don't see it in many other visceral tissues. That's why it's rare. And it's usually only 2 layers thick. Columnar, even rarer still, right? This is found in some areas of the back of the mouth, but we're not going to talk about the urethra. That's reproductive, the male urethra, which shares reproductive and urinary function. Some other glandular ducts. So you're not going to see this one too much. Just like transitional. Remember transitional? That's another rare one. This one here, that's another one. That's anything. If I ask you about transitional, think of the urinary organs, especially the bladder, the ureter. Urethra is, of course, like the male. But look, she said the same thing about the other. But there's many parts of the male urethra. Not many. There's 3. But the epithelium changes. So I would stick to the bladder, the transitional. Can we agree on that? So you never get it wrong. Easy points. So they change shape from cuboidal or columnar, not so much squamous. So we got to take a look at this. This is a hard one. This is aluminum, say the bladder. That could be the ureter too. Like here's the kidneys. Isn't that ironic that they're, or a consequence, like a coincidence that they're kidney shaped? But that joke went down cold. So this changes shape, like it goes from maybe thicker to thinner. And lumen is up here, so the cartoon doesn't tell me much, but this is transitional epithelium. When you look at it, you say, well, is that pseudostratified? No, there's no goblet cells. It's transitional. It's the basement membrane. So it depends on if the lumen is full or empty. It's really the change in that. So this is a difficult one. So basically, look at this picture and see if you can remember. Glands, we talked about glands. You have endocrine and exocrine. Please know the difference between those 2. And look at this unicellular gland cell. It's only 1 cell. So goblet is only 1 cell. It's not tissue. They're embedded in, especially that pseudostratified columnar epithelium and the simple columnar epithelium, if you could remember that. Craziness. So these are the glands with multicellular glands, and it's just epithelial tissue, and they're producing and secreting. Like this is an exocrine gland, like for possibly a sweat gland. Same here. And the duct, like this is the gland that's producing, and the duct will carry those products outside the body or into another duct. So this gland is exocrine because it's putting its products into a duct and then either out of the body. Endocrine, look at this one. Endocrine has blood supply, of course, tightly around it. So the hormones are going right into the blood and then they'll go to their targets. And you learn that in endocrine, AP2, but endocrine means into the blood. So there's blood vessels involved. So this is a great picture here. Ductless glands. So we're talking about, going through the interstitial space and going into target cells either by diffusion or transport or ligand gated changes. It depends, right? It depends. So if the hormone is a lipid like testosterone, which is a steroid, estrogen is a steroid, progesterone is a steroid, so they're lipids, and all steroids are based on cholesterol. Remember that when you take your test for chemistry, right? So the hormones leave the cell via that method of exocytosis, like a neurotransmitter is going to do. And then it goes through the interstitial fluid, then into the cell, easily if it's lipid soluble, like a steroid. But if it's a protein hormone, which is hydrophilic and non-lipid soluble, then it needs help getting its job done for whatever that wants to do, like insulin. Let's talk about insulin. Insulin is a hormone, endocrine. Insulin is a hormone that's produced and secreted by the pancreas, islets of Langerhans beta cells, in response to elevated blood glucose levels. So when insulin is released, it goes to other cells, and it's a protein. So it doesn't just walk right in like testosterone, because it's not lipid soluble. So it needs to bind to a receptor, a ligand gated situation here now. A ligand is the insulin to its insulin receptor. And a receptor is a protein, remember? So that helps insulin do its job. And what insulin's job is to open up the channel so glucose can go into the cell. And then glucose can go into the cytoplasm and be broken down to have its products be put into the mitochondria, which is the powerhouse of the cell, in the presence of oxygen to make ATP, aerobic respiration. So it's important to know how that happens and how to put all this together. So unicellular glands, I'm not going to ask you this, unicellular versus multicellular. Just know the concept of epithelial tissue and what it has to do with ducts and whatnot, right? Unicellular exocrine glands, like a goblet cell is a good example. It's just one cell, right? It's not goblet tissue. Look at this, a close-up of a goblet cell. Looks like a pimp cup or an upside-down bowling pin. Yeah. Craziness. But you should, as far as looking at a goblet cell, you should know what it looks like within that epithelial tissue, like we showed you before and last week. So I'm not that interested in all this. Tubuloalveolar glands. I'm not going to break those down. Compound, just what this means is you just have different branching types of glands. And again, this is more when you do digestive, when you do endocrine, where we talk about these. I think you have enough to worry about, right? This too, the types of secretion. Americrine is the exocytosis, like the pancreas, or sweat. So there's merocrine and which are eccrine glands, if you remember me showing that last week. But again, for the skin, all you have to say is sweat gland, because again, you have to know what a sebaceous gland is. That's holocrine, but you don't have to know that. You just have to be able to point it out and tell me what it produces for the lab and lecture. So listen up, yeah. Apocrine are also types of glands that kind of break off. Maybe mammary cells. I don't work in maybes in this class, so we don't have to worry about the difference in these, right? But maybe you should know eccrine, and we'll talk about that later when we do the integument. So some of the fragments actually break up, and go out, in holocrine glands, like part of it, like a sebaceous gland, that oily substance, and these get infected. These, like a sebaceous gland gets infected from your skin, right? You've seen that, then it gives you something like acne, and a lot of pus and all kinds of things happen, and blackheads usually have to do with the skin follicles and the sebaceous glands. Yeah. So any questions about epithelial tissue? That was a good review, I think. Alyssa, you okay? How do you stay awake so late? I don't know how you guys do it. You guys are amazing. So we'll go into connective tissue. We'll take a break. pretty soon. And again, all this should seem like a review. Does it all sound familiar? It just seems so long ago, right? Right, Gabrielle? This sounds familiar, though. We did this last week in a more generalized looking. So you're getting a couple of looks at it, even though we had that snow day, which was really a bummer. There's nothing good about that. So let's just move on a little bit then before we take a break. Tonight's a long one, I know. That's what happens. So connective tissue. So, I say so a lot. The most abundant and widely distributed tissue. So you should know what that protein is everywhere. That's collagen. Widespread. It's the most abundant protein in your body for sure. So the most abundant tissue is connective tissue. It's everywhere. It's binding, insulating, it's storing, it's protecting, transporting, and the blood. It doesn't do too much of that. It's the only thing the blood does. So you have the connective tissue proper, which is loose and dense. So remember, loose is that areolar, reticular adipose. Yeah. Dense is dense, irregular, or dense regular. And then you have dense irregular. Then you have elastic connective tissue, not elastic cartilage. Elastic connective tissue is categorized under dense. So here's the specialized ones. Cartilage, remember the cartilage? Remember my favorite, hyaline? And then there is the fibrocartilage. And then there's the elastic cartilage. Remember that one? The epiglottis. So remember, there's a difference between elastic connective tissue and elastic cartilage. I mean, elastic cartilage, of course, has elastic connective tissue in it, but there's only three places. Bone is a connective tissue. Blood is a connective tissue, because the definition of connective tissue is a tissue with sparse cells and a matrix. So it's different cells. That's what sparse means. Like blood is a connective tissue because you have the red blood cells, white blood cells, and platelets, and they're dispersed in a liquid matrix. Bone has your osteocytes, osteoblasts, osteoclasts, and you have a calcified matrix, and also a collagen protein matrix. So, and one of the loose ones is adipose, just one of the loose ones. Adipose is storing triglycerides, but it's a cell, adipocyte, with a gelatinous matrix, a fatty matrix, if you will. So we'll go through all those, and remember there's reticular. And I think the most common one of the loose of connective tissue is actually areolar. And dense is either dense regular connective tissue, like your tendons and ligaments. And then dense irregular connective tissue, which is basically your dermis of your skin. So a lot to remember, I know. So this breaks it down. I like tables. You know, before you open up Lab Practical, just go through, get the spelling, give yourself a nice chance. And again, I'm going to be pretty lenient on the Lab Practical with spelling, as long as you're in the right ballpark. So connective tissue proper, loose and dense. And here it breaks it down for you. Remember, elastic falls under dense. And here's your sparse cells, fibroblasts, which lay down the collagen, fibrocytes, mixed in cells, and your macrophages, right, your dense defense cells, sorry. And in loose connective tissue, the adipose sites filled with the triglycerides. So the matrix could be gel-like or thicker in collagen, like reticular fibers and elastic fibers. So the dense are very thick collagen fibers, except for elastic, of course. That's more elastic fibers. Make sense? So let's think of it this way. The denser the connective tissue, the thicker and more collagen fibers you have, because ultimately collagen is what gives it its strength and flexibility in connective tissue. Even bone. Bone has a lot of collagen. It's actually probably like 35 to 40% protein bone. Then 55% is all the hard mineralized calcium phosphate. So these are matrices within the tissue and around the cells. Okay, and some of the functions, you know, the resisting mechanical stress, it all makes sense. Of course, reservoir for water and salts, or energy, fat storage, and adipose. Because adipose, the lipid triglycerides, are used for stored energy. Stored energy. And here's the three cartilages. Know the difference between elastic cartilage and elastic connective tissue. Elastic connective tissue is what we saw in the aorta. Elastic cartilage is the ear, the epiglottis, and the eustachian tube or auditory tube. So chondro means cartilage, the prefix. Chondroblast is a immature cell producing, growing the matrix with collagen, really, which is in cartilage as well. Chondrocytes are the actual functional cells. Remember the chondrocytes in the lacunae, that little lake? We talked about that. Of course, they resist compression. All right? Cushioning, supporting. Not always connecting things. Yeah, that's really the tendons and ligaments. Remember, dense, regular connective tissue in tendons and ligaments. Tendons connect muscle to bone, or ligaments connect bone to bone. Really important. Okay, so here's like the more specialized we'll look at. Again, we're just going through the tables and giving an intro. So bone tissue is connective tissue, first of all. This is compact bone, and that's an easy one to see in histology. So tomorrow, for the FUs, the F group, group F troop coming in tomorrow, they'll probably see this one easier than most, compact bone. Spongy bone is different. That's trabecula, where compact bone is the osteon, like we talked about in the lab. Osteoblasts, so osteo prefix is bone. Osteocytes are the functional resident cell, where blasts are the more immature cells, and they're laying down collagen. Because I think I told you that all bone is made from cartilage. So it starts out as cartilage, which is mostly collagen, and then calcifies to become hard bone, even this in a long bone especially. So we're going to go crazy talking about that next time. Hard tissue, supportive tissue, protective tissue, bone, compact bone. Blood is that liquid matrix with you can see the red blood cell doesn't have a nucleus, so it looks like just a red disc or clear disc. Doesn't have a nucleus. Red blood cells are anuclear. They're also called erythrocytes. White blood cells are leukocytes. They have big, weird-looking nuclei, actually. So in AP2, you really study the white blood cells. What's the difference between a neutrophil and an eosinophil, or a phil for a modern family, whatever phils are involved. And then we have platelets, which, by the way, platelets are also anuclear. Cool. So blood is a fluid tissue that functions to carry oxygen, nutrients, carbon dioxide, and other chemical messengers like the hormones. So again, we're not doing that in AP1, but you should know the function, and you should know that it's a connective tissue right here, right now. That's really important. So characteristics, different characteristics make different connective tissues different from each other, obviously. I think I mentioned they all arise from this stem tissue called mesenchyme. Now, mesenchyme already decided it's going to be connective tissue, but all connective tissues start, including blood, with mesenchyme. Tissue origin. So again, on Friday night, you know, you have the sperm swims up the fallopian tube from the uterus and then meets up with the oocyte in the fallopian tube. They form one cell, and then all the tissues start. So in less than 8 weeks, or much less than 8 weeks, some of that tissue that's developing from all that mitosis is going to say, okay, I'm going to be the mesenchyme of Jeremiah. I'm going to build his connective tissue. And then from there it starts. So, and the mesenchyme is basically going to develop into collagen, really, right? So they're pretty vascular. connective tissue is pretty vascular especially compared to epithelial tissue. Although remember this, I might have mentioned this on Thursday, that dense regular connective tissue like ligaments and tendons and that fascia connects the muscle to muscle, that is not very vascular. That's very avascular actually. So I can't say every single connective tissue is vascular, but most of it is. The dermis of our skin is very vascular and innervated too. Like innervated is, like we mentioned before, is nerve supply, either sensory or motor. Sensory is like the pain receptors or the thermoreceptors. When you feel pain, you feel temperatures, cold and hot, you feel tickle, you can feel pressure. All those things are receptors, sensory. Motor is more going to a muscle to make it contract, like the erector pili muscle. Remember that? The erector pili muscle contracts and gives you goose flesh. It's smooth muscle, by the way, and that's what I'm going to ask you on the test. So the extracellular matrix is basically the matrix that I'm talking about. Extra means outside the cell, like the E. I don't want to say ECF because it's not all fluid anymore. So it's outside the cell. So remember, the definition of connective tissue, which starts as mesenchyme, is sparse cells with an extracellular matrix. So it depends on what the matrix is. Can it bear weight and withstand tension? Endure abuse. This is abuse. 3-hour lecture is abuse. Our connective tissue better be ready. Okay, so now we have other main elements that it has. So ground substance is basically just part of the matrix. Like you might have other types of tissue in there, like little granules and cells too. Cells, which could be macrophages. Let me just tell you what they are. There's one called macrophage. And we know what a macrophage is. That's a big eater, right? That's an immune cell. So that's important. And in the skin, do you remember what we call the macrophage? We called it a Langerhans cell. Wasn't that weird? Langerhans cell. Does anybody have a question? I can't see. I see something flashing. Sorry, it'd be a nice time to take a little question. Anybody have a question? Or you can't hear something? What is it? Oh, thank you, Anthony. So there's another cell called a mast cell. You see this in the skin too. A mast cell produces that chemical called histamine. So the ground substance is like granules that can be like little chemicals that produce different effects. It could be little fat globules, something like that. But the fibers are those like reticular fibers. Structure-ish. Or you could have elastic fibers too. Depends on where the tissue is. So when you see areolar connective tissue, you can see that's a perfect example of all these things. That's like the mother of all connective tissues, and it's loose. Remember, reticular, areolar is loose connective tissue. Areolar is loose. So is reticular, and so is adipose. So get those classifications down to easy points. Look at those tables before you go in. You'll be golden. So ground substance and fibers. And how they're arranged, like are the collagen fibers really thick and close together, or they kind of dispersed and you have a cell, you have some wiry reticular. It looks like a mishmash, something you would see at the Whitney. Yeah, it looks like the guy's face. See that? So you have to use your imagination, especially for 3 hours on a Wednesday night. So unstructured gel-like connective tissue, it'll go on and on, but again, for lab, you have to visualize it. So I think that always helps. You know, ECF is interstitial fluid. That's between cells. Adhesion proteins are more for attaching cells. And proteoglycans, don't worry about that, but that we talked about when we did the cell membrane, right? The glycocalyx. And one example of proteoglycan is a high hyaluronic acid, which is basically a building block for cartilage, or chondroitin sulfate. If you guys are into your holistic medicine or something, or if you're Kylie Jenner making billions of dollars on this stuff, these are building blocks for cartilage, really, and not only cartilage, but for other connective tissues. So they used to sell this stuff like for your joints to build up your cartilage. So it's basically a build-up product of the mesenchyme. Best way I could say it for you. And there is some water. And there's water in bone, too. There's very trace amounts. So sometimes water is part of the ground substance. Of course, it's a big part of plasma. Like any fluid in your body is major water component. So connective tissue, 3 types of fibers. Here's your collagen. That's the strongest. That's the, the I-beams, the heavy steel of the building, and most abundant. And it's protein. It doesn't even tell you that. I'm telling you right now, this is protein. Classic protein. Has to be transcribed from your DNA to messenger RNA and built on a ribosome. Specific amino acids. That's a machinery that has to work right. And it's tough. And it's kind of flexible too. Tensile strength. It'll bend but not break, hopefully. And we do have collagen in our bones. It's the protein in your bones, which again is connective tissue. Elastic fibers allow for that compliance, stretch, and recoil back to its original shape. Elastin is a protein, actually. That's a cool thing. Elastin is a protein involved in those fibers. So that has to be transcribed in the DNA, translated on the ribosome into a protein. Reticular are a little skinnier than your collagen fibers, but they really do look like elastic, so be careful there. They branch like crazy. And of course, different chemistry, which means it's not so much high hyaluronic acid, that you don't care about that. And it's structure, but it allows for more give. Like if you're building a bridge that's exposed to high winds, I would use reticular fibers. Collagen may not be as bendable, even though it's strong. So it all has a reason to be there. Here's some cells now. Blast cells, I tell you, remember blast is always immature. And they're usually secreting something like collagen. Fibroblasts, anyway, secrete collagen, pretty much. Chondroblasts, secreting collagen. Osteoblasts, secreting collagen. I'll tell you right now, they're making protein, specialized machinery inside these cells. So you'll see nuclei of fibroblasts. Chondroblasts, I don't think you're going to see that a lot in your histology. Or osteoblasts. We'll talk about osteoblasts when we do skeletal and lecture. Crazy. Hematopoiesis, we're going to talk about that. That's making blood cells, and that all happens in the bone marrow, the red bone marrow too. So that's like stem cells. That is stem cells. But of course, these are bone-making, blood-making cells. These are bone-making cells. These are cartilage-making cells. These are more connective tissue-making cells. But it's all about making collagen. Yeah, so mature cells are called cytes. Blasts are immature. Cool. So it's a site. It's a prefix, suffix. And that goes for any tissues really, except for like the, I don't think we ever use CYTE in the nervous system. It's always just neuron or glial cell. Like we can call a muscle a myocyte, but we don't call a neuron a neurocyte or anything like that, just so you know. Because you have to learn this speech, this whole terminology, this language. We spent a lot of time in the first lab just learning new words and all the exercises you have to do. Okay, so other types of cells, you have fat cells, adipocytes, they store triglycerides for long-term energy. Then you have your leukocytes, and these are examples, two of them here. There's 5 total. There's also basophils and monocytes, along with this group. And they're about immunity, or they respond to injured tissue. So this is where you get this word, and pretty much all of these things. Listen to this word. This is really important. I know there's a lot of nuts and bolts here, but this is something you should talk about. When something's injured, you're going to get this response, and this is called inflammation. So you're going to get white blood cells, and these are from the blood. Then you have these things called mast cells, and that produces histamine, which histamine is a big part of inflammation. Like, you know, when you have too much inflammation, you get stung by a bee or you're allergic to something, and your body goes into this crazy immune response to the point where you might not be able to breathe. And you'd have to take an antihistamine to stop that production. So mast cells, remember, produce histamine, which is a big part of inflammation. Immune response against something foreign or an allergen, right? If you're allergic to something specific, like grass or dust or dander or peanuts, whatever. Macrophages, you know what they are. So these are in the tissue. These are tissue cells. These are blood cells. So phagocytes are eating cells. Macro means big eaters. So they eat up like problems with dead cells or dysfunctional cells or bad DNA or bad proteins, anything that's not functional that can cause a problem. Microorganisms are like a bad bacteria, virus, parasite, something like that. So it's very immune response, and it all adds up to this inflammatory response within your tissue. All right, what are we looking at? A prototype. A prototype. Wow, this is great. So this has everything, but if you really look at this, this is areolar connective tissue. So this is loose connective tissue proper. So you see different cells, and make sure you can see all that. So the thick, again, this is a cartoon, so you have to look at real histology. You see the thick fibers are collagen. Then you have this fibroblast with a nucleus that lays down the collagen. Then you have these little inclusions, whatever they called it, like little fat cells within the tissue. And then you have reticular fibers of these blue ones, and elastic fibers are a little thicker than the reticular in this case. Here's a mast cell. Remember, a mast cell is going to release histamine if this tissue is damaged in response to protect the area, to protect the blood cells, really. I mean, this is vascular. So you got this big blood vessel running through this. This is vascular. This is a white blood cell, like a neutrophil. And here's a macrophage. Looks like a fibroblast, doesn't it? So this is a macrophage. And lymphocyte's another white blood cell. And so the neutrophil, I might have told you this last week, it's more about going after and eating bacteria. It's not a bacteria, it's against bacteria. And lymphocyte is more against or anti-virus or cancer. Yeah, so neutrophil is like a little eater. Macrophage is the big eater. So if this happens, and histamine is a job, like a histamine of these granules in here. So let's say this tissue is injured in whatever, a cut, an acid, a bee sting, you had surgery, whatever it is. These granules of histamine are going to be released, and some of them are going to go into the blood. It's like a signal that tells more inflammatory cells, like more macrophages, bring in more neutrophils, bring in more lymphocytes, bring in more mast cells, bring platelets, whatever you can do to preserve and fix this injured tissue. So they're chemical messengers in a way. And these are specialized. The cartilage, bone, and blood are specialized. So connective tissue proper, loose, and dense. And then subcategories. Look how nice this is. This is a perfect thing. This is the second thing you should look at. Okay, remember these. Go in here. It's hard to practice, like you know, because we're not in lab every week, so you really have to rely on this, and you're not really using Mastering A&P, which is like an online lab. So I mean, you could if you have it. It's good practice. It just gives you like little quizzes and helps you study sometimes. Just put all this stuff together. So connective tissue proper is split into loose and dense. And then you have the specialized, which are cartilage. You should know the three cartilages and where they are and what their functions are. All of them you should know where, what they look like, and their functions all the way through. And then bone and blood. Bone and blood are probably the easiest to recognize of the connective tissues when you see the histology. Yeah, connective tissue proper is everything except for the specialized. bone, blood, and cartilage. So areolar is the biggest one. So that's another thing. You remember that, areolar. Pretty much what we looked at already. So what does it do? It supports and binds, right? And it packs material like, other tissue really. It kind of holds it together. So it has fibroblasts that you can see, and that's producing the collagen. And then you have other things like reticular fibers too, and some little fat cells and macrophages and mast cells. Basically what we looked at. So this is connective tissue proper. This is your areolar connective tissue. Where do we find it? Under epithelium. So it's kind of connecting, the epithelium to other structures like organs, or holding capillaries to it, holding things together. So it's part of the bottom of the epithelium, which is important, below the basement membrane. So look at the tissue now. This almost doesn't look real. It looks like a mistake you saw in a microscope. You see the elastic fibers. You can see the ground substance in between. And then the fibroblast nucleus. And then if you look at these, this is the collagen fibers, this thick pinkish staining. So all this is all nuclei of fibroblasts. But you should be able to identify it. That's number one. This is loose connective tissue, and it's areolar. It's not adipose, right? And it's not reticular. They look different. Adipose, nothing looks like this. Adipose, especially the white fat, and you look at the microscope like we were looking at it, or when you look at it, it kind of burns your eyes. It's so light. Light comes through and gets you, like I said. So the cells have nuclei, and they're called adipocytes, and they have a lot of blood supply. So they function to store energy and insulate other organs, and a shock absorber. Makes sense, right? So that's the fat you're going to see. Brown fat, which as adults, we don't have so much of this anymore. So this is good for using lipids to produce ATP within the fat. So this is really healthy, brown fat. If you have more of that when you're an adult, it's definitely good. It's not a problem. So here's adipose. It's showing it in the adipose tissue in the breast mammary glands, but it's pretty much behind the eyes. It's in between. It's around your heart normally. Not in your brain though. There's no fat padding there. You have cerebrospinal fluid there. So it kind of acts as insulators in different areas. Look at this. You see the blood cell going through there. That's pretty cool. This is a capillary. So I don't know if you noticed that. That's not part of the cell. So I just noticed that myself. These are good slides. So you see the big vacuoles. And this is all cytoplasm where you'd have the triglycerides being stored. And here's the membrane. Not the basement membrane, but the cell membrane. And here's the nucleus of the adipocyte to have the triglycerides within there. So this is what you see, and it doesn't look like anything else. So it should be fairly simple to go through that. So reticular is loose connective tissue proper, and it resembles areolar a bit because of the fibers, but the fibroblasts are called reticular cells. So we have to look at that. Stroma is like a lattice, like the protein supportive structure of something. Right? We mentioned this in lab. So reticular fibers form that supportive structure, like say around the kidney or the spleen, it says here. So holding it in place and kind of separating it from other lobes, like the lungs has stroma, reticular fibers that kind of separate the lobes, if you will. The liver, same thing. So organs, supportive and holds it together. So there's nothing looks like this either. So again, it's showing you the spleen down there. The spleen's only on the, remember, spleen is in the upper left quadrant. That's a question like on a lab practical, like what's in the upper left quadrant. This is the spleen is up there. I just remember a lot of students getting that wrong over the years because they forgot where the spleen is. Just want to throw that at you. So this is, this reticular fiber is a little thick, in this picture. And it stains darker. Again, this is something we have to look at in the lab, but this is all you have. So you have to really look at that. And there's some sparse cells like a white blood cell in there. So this is reticular, which falls under the loose connective tissue with adipose and areolar. So it kind of looks like elastic cartilage too. I'll show you elastic cartilage today too, you'll see. So here's the CT proper dense connective tissues. Dense regular, remember dense regular is your ligaments, connects bone to bone. Your tendons that connect muscle to muscle. And fascia, or also known as aponeurosis, connects the muscle to muscle. And it's not fat, it's protein, it's dense, regular connective tissue. So it continues all the way through the muscle fibers, which you'll learn in muscle, how this fascia is kind of an extension of a tendon into the muscle itself. But those are what you should remember for dense regular. And this is very avascular. It's the only connective tissue that's this avascular. So if you injure a tendon or a ligament, it takes much longer to heal because of that lack of vascularity. Dense irregular, and you should know what they both look like, is more in the dermis of the skin. The dermis, not the epidermis. The epidermis is epithelial tissue. The hypodermis is more loose connective tissue like adipose, but there's also areolar there too. And then your elastic connective tissue, elastic CT, not elastic cartilage. This is the one you see in the blood vessel walls like the aorta. The aorta is working under high pressure because the left ventricle of the heart is pushing blood into it at very high pressure. Okay, dense connective tissue, high tensile strength. That's what it's for. Protection of your joints, really. Closely packed, resistant. And the fibers do look wavy. So I remember I told you last week, when you look at dense regular connective tissue, sometimes it looks like smooth muscle, which is a completely different tissue. Muscle is not connective tissue, you know that. So it does look kind of wavy. We'll take a look at that. And there's nuclei that you see in there, because the wavy tissue is collagen. That's wavy. That's like a wavy, thick collagen. And you'll see nuclei, but the nuclei, of course, it'll be different stain, is fibroblast nuclei. Very few cells, so it's not so many cells, not as many sparse cells. And like I said, poor blood supply. So a perfect example is tendons and ligaments and that fascia that's really an extension of a tendon between the muscles. There it is. What can I tell you? There it is. It's ligament, shoulder, knee, elbow, ankle. What else? Metacarpal phalange. All the joints that have ligaments have dense, regular connective tissue, which makes up those ligaments or tendons. Right? So the tendons connecting the muscle to the bone. You have to learn all those bone markings, but I'm not going to bring that up right now. We got enough to worry about. Dense, irregular connective tissue. It's not as dense. There's more space between those collagen fibers. That's the best I can give you without looking at it. So the dermis, below the epidermis, it's all dense, irregular connective tissue. Get used to saying it. And the joint capsules, you'll see this with especially these joints called synovial joints. Get used to this word, synovial joints. Articulations, right? Joints are called articulations. Articulations are connections of bones. So some of these synovial joints, which are very movable joints, like your shoulder, your elbow, your hip, your knee, your ankle, they're all movable joints, some more than others. The shoulder is the most movable joint, so it has a nice, thick, dense, irregular fibrous joint capsule. And the hip has one too. So it's like a rubbery ace bandage that you put around the joints, the joint capsule. And it's a common feature in synovial joints, which also has synovial fluid. There's a whole thing we're going to learn about synovial joints, and that's the most one I'm going to talk about. So fibrous coverings of some organs, like some organs are encapsulated, like the kidney is encapsulated as a capsule, like a tomato skin, right? It has that capsulation, and usually those coverings, connective tissue coverings, are dense, irregular connective tissue. Like the epicardium of the heart is dense, irregular connective tissue. So if you ordered a heart or a kidney on Amazon, it would come nicely wrapped in dense, irregular packaging, connective tissue. So this could be red, right? This is dense, irregular, but you could see the collagen fibers could be red, not blue. It just depends on what stain we used. So I think this is different, right? It looks different than dense, regular. I did this the other night. Dense, regular, dense, irregular. There's more room in between. Dense, regular, dense, irregular. Collagen. Collagen. How many times did I say it? 80 times, Jeremiah, last time? Connective tissue proper, continuing with dense. Now we have elastic connective tissue. Remember, this is not elastic cartilage. Some ligaments are more elastic than others, right? We'll learn about that, some ligaments, but I like it here. Large artery. The largest artery in your body is the aorta. It's huge. It's a roadway for blood, which we know what that contains. It's an interstate highway like the New Jersey Turnpike, Route 95. It's not like Route 10, Route 18. Those are arteries. So they need some recoil and conformity and compliance to push, or have that blood being pushed at it. So they have to have that resistance. So here it is. Here's the aorta. It's coming up. I can see it there. I just can't click on it. So here's the lumen, where the blood is. Blood. Another connective tissue. Connective tissue everywhere. Here's the aorta coming out of the heart. So the blood is coming out of the heart. So by definition, an aorta is an artery. The definition of an artery is a vessel that takes blood away from the heart or out of the heart. Definition of a vein is a vessel that drains blood back into the heart, brings blood back. So arteries out of the heart, veins back in. And there it is. There's the picture. A lot of elastic fibers. And they look kind of windy. They're a little wavy too. So note the lumen, right? Where the red blood cell. Remember, red blood cell has no nucleus. Then you have white blood cells with a big nucleus, like a lymphocyte. Platelet's a little fragmented cell with no nucleus. Leukocyte, erythrocyte, and thrombocyte. Know the big boy terms, big girl terms. So now the last specialized one is the cartilage. Not the last, we still got to talk about bone. So this is the chondro, like chondrocytes. So chondroblasts are laying down the matrix. They're immature during growth of that tissue. Right? Chondrocytes are found in those little cribs, cavities, lakes. Lacunae means lake. Right? So a lot of water packed between these packed collagen fibers, and the chondroitin and hyaluronic acid, which is going to save your joints if you buy it at Costco by the dozen, or Kylie Jenner sells it to you. It's tough yet flexible. That's like your professor needs to be tough yet flexible, like cartilage. But avascular, we don't want to be that. So it's avascular. Avascular. So all the cartilage is avascular. You hearing this? It has an outer covering too. It's not a capsule. It's just called perichondrium. Kind of like the periosteum, which you'll learn about around the bone. It actually is a little more connective tissue. So the perichondrium gives rise to the cells. So that's kind of confusing. I think we're going to save that particular concept for bone. We've got to talk you through that, starting with the periosteum. Then we'll talk about the perichondrium. Peri. 3 types of cartilages. Get it right. Hyaline. Spell it right. Most abundant, the gristle connective tissue, I guess. I don't know. I guess in steak. I think that's more fascia, but that's okay. Appears shiny, bluish, because like this is the femur head, right? Femur head. And this is the other femur head, and this is the tibia down here. So the femur proximally, epiphysis, and the tibia distally form the knee joint. So the ends of all joints, let's get this down now, why not? The end of all joints has a remnant of the cartilage model kind of on the top of it, right? And that's called articular cartilage. And it's usually blue and shiny. So those ends of the bones are covered with hyaline cartilage, and that's called articular cartilage. Tips of long bones. So that, we'll talk about that with joints too. But you also see it in rings around the trachea, your voice box, the larynx, and your costocartilage, which you're going to learn very soon. I think I showed you that in lab. Costocartilage. The two labs we had. Okay. Elastic cartilage, not just elastic connective tissue, is your ears, the pinna of your ears, and your epiglottis, and your auditory tube, also known as the eustachian tube. Fibrocartilage, and you should know where this is. This is and what it looks like, and it looks a little different. This is confusing too. So it's kind of between dense regular connective tissue and hyaline, but that doesn't really show up in the picture. So this is between your discs. I'm sorry, between your vertebrae, intervertebral discs, your IVD. And in your knee, you have little discs called menisci. And between the two pubic bones, you have the pubic symphysis. I definitely showed you that. Pubic symphysis. That's always an answer on the practical. Symphysis. There you go. Spell it right. Type it right, right? This is in this day and age. What do we do before Blackboard exams? I don't even remember anymore. So here's hyaline. This one's definitely something I would show you. So you see the dark staining chondrocytes in the little lakey lacunae. This is all collagen matrix. So here's the costocartilage, here's the ribs, here's the sternum. And what the cartilage is doing, besides giving strength and flexibility to the thoracic cage, it connects the ribs to the sternum. Crazy stuff. This is elastic cartilage now, not just elastic connective tissue. That's elastic cartilage. Where is it? Where'd it go? What happened? Did I just erase that? That's fibrocartilage. There you go. So this really doesn't look like anything else. You see fibers. You know this is not epithelial tissue. And here's one of the things you got to be careful with here. I got to stay on this for a second. This kind of, and again, if I put the two slides next to each other, you'd say no. But this kind of looks like nervous tissue a little bit. And only because of the stringy look and the cell bodies, but these are chondrocytes and lacunae. This is all elastic fibers. So these are the three you should get to know next to each other, or make sure you can differentiate between reticular connective tissue, elastic cartilage, and nervous tissue. Yeah, I think, and once you look at it, you see that it's different histologically. So this is fibrocartilage, like in intervertebral discs. So yeah, nothing really looks like this. It kind of looks like loose irregular, I think, too. So we have to go through that, and we'll look at the slides tomorrow and get a good look at it and kind of draw it and go through the big, heavy, expensive lab manual. Pictures in there are great too to prepare you for the lab anyway, because now we're just looking and kind of reviewing, which is really good. This is really good that this is working out as far as the timing goes. So get this first exam out of the way and then start the practical, or however you want to do it. Okay. So let's move on to, maybe we're going to go to bone now. Okay. Bone tissue. Osseous is bone. know the functions, protection. Has some fat storage too. The triglycerides are stored within the cavities of bone. And it has a lot of collagen. And there's always cartilage involved with bone. In fact, bone comes from cartilage. And the inorganic salts are the hard matrix, mostly calcium and phosphate too. So osteoblasts are laying down the matrix. And the matrix really starts with one thing, and it's collagen. So of course, remember that bone is a connective tissue, and it's derived from mesenchymal tissue. So the osteocytes are the maintaining cells, the resident cells, and they're in lacunae too. We saw chondrocytes in lacunae back in the cartilage, and now we're seeing, remember, chondro means cartilage. Cartilage, you have to get the difference. I know that is a tough thing. I still have trouble with that. That I say connective tissue, and I should be saying cartilage. I say cartilage when I should be saying connective tissue. So chondro means cartilage. Osteo means bone. That's all connective tissue. I just messed you up, didn't I? So osteon is like an individual structure within compact bone. So an osteon is like a unit of compact bone, the hard bone, the bone that's kind of lining the outside of your femur, right, is compact bone, or your humerus, or your tibia, fibula, ulna, and radius. So there it is. And this is a humerus. Picture the humerus down here. Here's the cartoon. Here's the humerus, which in a couple of labs, you'll be able to tell me what the humerus is compared to the femur. So these are the osteons, these circular rings in compact bone only. So you have osteocytes, or the little black things that look like ants, and they're in lacunae. Lamella are the layers. And this is what it looks like. Nothing looks like this. Central canal is how the nutrients come in, upward and outward. And then they have these little canaliculi, which kind of feed the osteocytes from the central canal to the osteocytes. Canaliculi to the lacunae. It's a really nice setup. It's a nice little living arrangement for the osteocytes. Make sure I don't say chondrocytes, right? Blood, atypical, because it's nothing like the rest of the connective tissues. That makes sense. You're probably surprised to hear that blood is a connective tissue. I was. Right, so you have erythrocytes, or the red blood cells. Platelets, or thrombocytes for clotting. Red blood cells, no nucleus. Anuclear, just for carrying oxygen. Platelets for clotting. The white blood cells are called leukocytes, and therefore immunity. So blood has two things. It has formed elements, which are basically the cells, which I just told you, erythrocyte, thrombocyte, and leukocyte. Then they have a fluid matrix. And the fluid matrix is the plasma. So it's formed elements plus a liquid matrix, which is plasma. I would say liquid matrix. And the plasma is mostly water anyway. So it's the water is carrying nutrients, carbohydrates like glucose, your amino acids, your fatty acids, and wastes like carbon dioxide or urea, other breakdown products, gases like oxygen, nitrogen, carbon dioxide, and other substances like hormones, chemical messengers, and proteins, blood proteins, really important. So nothing looks like this. And this is blood that's within blood vessels. That's really all you have to say. So all this white is the plasma. And these are anuclear. There's no nucleus. That's why there's no dark staining. Because all these cells do is carry oxygen. And this is a large nucleated lymphocyte, which is a white blood cell, a leukocyte. Here's a bacteria-fighting neutrophil. But there are others. There's basophil, there's eosinophil, and monocyte. Okay, and this could be either a fragmented cell or a platelet. It stains dark, though. It does stain dark. So when you're looking at a microscope, you see like little dots and stuff. You got to be careful that what you're looking at is not an artifact, which is just like somebody dropped something on the slide or there's something got into the tissue that really shouldn't be there. An artifact. That means it's not really what you're looking at. So now we move on to another tissue, and we're not going to go deep into this, but you have to know this, what muscle looks like versus nerve versus connective tissue and epithelial tissue. So skeletal muscle is voluntary. It's the only one that's voluntary. The other are involuntary, cardiac, and smooth. Cardiac, smooth muscle is also called the visceral muscle. Okay, so skeletal muscle is also striated, but so is cardiac muscle. Smooth muscle is unstriated, but also involuntary. And we'll show you other things. So skeletal muscle is the muscle you're going to learn that's voluntary, that moves bones and joints. Cardiac muscle is only the heart. Only the heart. Smooth muscle is all the muscle in your linings, not the linings, but the walls of your gastrointestinal organs, your esophagus, your stomach, your small and large intestine, your reproductive tubes, but also in blood vessel walls. So you not only have elastic tissue in there, you have smooth muscle. So remember the erector pili. I'll tell you that right now. It's a question on the exam, practical. The erector pili, the goosebump muscle, is smooth muscle, not skeletal, and it's involuntary. So let's take a look. And know the appearance of it, and I'll go through that with you. Like skeletal muscle is multinucleated, and the nuclei is in the periphery. I know I did this last week. And it's very linear looking. And of course, it's striated and it's voluntary, like here showing the triceps brachii. So it's got a lot of mitochondria too, but you don't see that. You just see the multinucleated peripheral nuclei. Straight lines. Now, I'm not talking about the striations. I'm talking about the way the cell is arranged and packaged. Very straight lined. Of course, it's voluntary. Cardiac muscle, and the big thing here, I'm not going to quiz you on it because I know you guys are getting tired, but intercalated discs are the hallmark of cardiac muscle, involuntary. It's definitely striated. The nucleus is usually central located nucleus, one usually, sometimes two, and the cells are branched. So you have like these little fibers that are more electrical fibers in between, and of course blood vessels in between. Yeah, but these are Purkinje cells, which we'll learn later. But this is cardiac, involuntary, branched in the heart, and it pumps blood. That's the answer on the practical. Pumps blood out, and there you go. Smooth muscle, spindle-shaped muscles. And here we go. So be careful with this one and the dense regular connective tissue. Spindle-shaped nuclei, that's cool. Or there's a different word for it. The cells are not branched, they're pretty straight, but the cells are actually spindle-shaped. There's no striations. So remember, cardiac muscle, because this is another answer. Let me just give you some answers while you guys are half asleep. Intercalated discs, you have to know that. And that's cardiac, or heart muscle. So it pumps blood, right, from the myocardium into the aorta, which is right here with all the elastic connective tissue. Involuntary, no visible fibers. So here's another one. This is good. It shows you another one in the intestinal walls. Yeah, same one. Sorry, must have jumped ahead. Same one. So now nervous tissue. So be careful with this one and elastic cartilage because it kind of stains the same. So here's the cell body of a neuron. All these wires are axons. They're axons, or you could say nerve fibers. I'd be okay with that on this test. But later on in the semester, we're going to live this stuff. So, and all the nuclei you see out here are glial cell nuclei. Glial cell nuclei. Might as well get it down now. They're not neurons. It was a supportive cell. So your coverings and membranes, remember you have cutaneous membranes or skin. Mucous membranes are inside the tubes with the endothelial cells, usually, or the mucus-producing cells like goblet cells interspersed. Serous membranes are between the organs and the cavities. So if they line the cavities and they line the outer portion of the actual organs. So cutaneous is skin. It's another name for skin. Epidermis is epithelial tissue. And it's stratified squamous. You got to learn again. And the dermis is dense, mostly dense, irregular connective tissue. Proper. Unlike other membranes, the cutaneous is a dry membrane. Okay. Cutaneous membrane, just showing the outer covering of the skin, seen with the naked eye. So there is a layer called mucosa, which produces the mucus, and usually that's in those same areas that are producing mucus, like inside the tubes of the digestive system, reproductive system as well, and respiratory. Okay, and loose connective tissue like the areolar connective tissue is sometimes referred to as the lamina propria of the membrane, or the covering. So now we're kind of mixing tissues now a little bit. We didn't get to organ systems yet, but we're still talking about mostly the epithelial connection with connective tissue. So the mucosa, of course, there's going to be a lot of goblet cells in that area, in the mouth, and the esophagus, digestive, and the respiratory bronchi. Serosa is between the cavity and the organ. And most of these, like we said before, remember mesothelium and endothelium, is simple squamous epithelium. So it lines the cavities. Now we're talking about cavities. Remember the cavities you have to know for lab, of course, the dorsal cavities, the cranium, vertebral, and then the thoracic, abdominal, pelvic cavities. So they're lined with this serosa. And serous fluid is the fluid that's between them, not mucus, between the cavities. So around the lungs, it's known as pleura. You can lose the E, it'd be okay. Around the heart, it's called pericardium, the cavities, too. And around the abdominals, it's called peritoneum. So this is actually important for lab, too, that you know pleura means lungs, pericardium, cardio means heart, and then peritoneum, or peritoneum, however you want to say it, is the abdominal area. So here's, going through, we did this in lab, again, parietal and visceral, the difference, like this is a better one to see, I think, use the pericardium and the pleural. So the visceral pleura lines the lungs, the parietal pleura lines the inner part of the thoracic cavity. So parietal versus visceral. Remember, visceral means organ. So that's usually lining the organ, the outside of the organ. Parietal is outside. Same thing down here in the peritoneum. Tissue repair. So let's finish this up and take a break. Okay. So now the tissue has been injured. So now we need inflammatory response, which in this case is completely appropriate. And the immune response is part of that inflammatory. And let's get this repair done very quickly. Okay, so I'm just going to kind of walk you through this. Okay, occurs in two major ways. First, regeneration, and the same kind of tissue comes in. So this is good, but after a while, you get what's really like scar tissue. So the word fibrosis is scar tissue. So it's a lot of collagen, really. It's connective tissue that kind of not destroy, it replaces the tissue that's been injured. That's why you get scarring, because of this fibrosis. So regeneration is good, like the beginning of the healing looks nice and soft and okay, but after a while it just gets harder and harder because of the connective tissue that's put in. It's like the body brings in a really cheap material after tissue has been injured. So here's the steps. Let's stay with me here. Stay with me here. I know I'm recording, which is great. So the steps of tissue repair. Step 1, just say inflammation. Always say that. Tissue repair, not bone fracture repair. Tissue repair. The first step is inflammation. And that's inflammatory cells like macrophages, mast cells, and other white blood cells that come in. The blood vessels actually open up to bring in more blood, bring in more cells. Remember, those granules, those histamine chemicals will open up the blood vessels. You'll get vasodilation, that's called. That's where you get all the redness and the heat and the itchiness and the pain from the dilation of blood vessels, more blood to the area. So the blood vessels become more permeable. You should know what permeable means, moving things in and out of a membrane. In this case, the endothelium of the capillaries. And platelets come in, so blood clotting will occur within this stage of inflammation. Now, this is completely appropriate when you're injured, but say this is happening in your blood vessels all the time because you're eating at Buffalo Wild Wings too much. This could affect the inner linings of your blood vessels, inflammation, but completely appropriate. So step 1, inflammation, and all those good things that come through there. So inflammation sets the stage. Let's see if we can do this as a slideshow. Maybe it'll look better. If it's worth it, let's see. So step 1 is inflammation. You can see what's going on here. Tissue is injured down into the dermis. Like this, you should remember this is the epidermis, into the dermis where the blood supply is. And here's the blood vessel bringing in white blood cells. Veins drain, arteries deliver, feed. Arteries from the heart, veins back to the heart. So trauma, so you're having this response. Step 2, organization restores the blood supply. So, of course, blood clotting, platelets have a lot to do with that. New tissue comes in. Now, remember, again, the slideshow is not as good as I thought it would be. The blood vessels in this area, like capillaries, are endothelium mostly, so they're able to regenerate. And epithelium is very mitotic, so it could regenerate fast. And the fibroblasts begin to produce collagen. So the first step is inflammation. The second step is organization, restoring the blood supply. Because you need the stuff from the blood, the nutrients, the cells, and the other chemicals that help that blood repair, or that tissue repair, sorry. So again, you can see it now. You see what's filling in to that area. Granulation tissue, which is mostly collagen. Fibroblasts laying down the collagen. The macrophages building up, left over from the inflammation. And then step 3 is the regeneration of the fibrous effect and permanent repair. So now we're getting scabby tissue. Fibrous tissue is maturing, starting to get a scar tissue. And that's step 3. So now we're nice and closed up, but you're left with that, not cartilage, you have basically connective tissue in there. It's fibrosis. So there you go. So remember those. Just the first step is inflammation. That's basically what I'm going to ask you. The first step is inflammation. The last step is the fibrosing. So let me just go through that one more time. So make sure inflammation, organization of the blood supply and regeneration, and then regeneration of the fibrosis, which is the scar tissue. Your book kind of complicates it more than it really should be. So it depends on which tissues are injured, right? What if a ligament gets injured with no vascularity? That takes a really crazy long time, and it needs physiotherapy. It needs all kinds of help. But the loose tissue, like the loose tissue, well, and I shouldn't say loose, I'm not just talking about connective tissue. Epithelial tissue we know is highly regenerative. Areolar connective tissue's got a lot of blood supply, very good. dense irregular tissue much better than dense regular. And the blood-forming tissue, like the hematopoietic bone marrow. But the ones with moderate, smooth muscle, they can regenerate. But I'll tell you this, the least is cardiac muscle for regeneration. So smooth muscle is the most regenerative of the three muscle types. Yeah, so dense regular, not good. Not good at all. So no regenerative ability is cardiac muscle, nervous tissue, right? Brain and spinal cord. So you injure a cardiac muscle, it's not coming back. That's my takeaway here for this. So the most regenerative muscle is smooth muscle. The least regenerating muscle type is cardiac muscle, which is kind of scary if somebody has some type of heart attack, myocardial infarction, right? It's not coming back. So you really have to rely on the other muscle cells of the heart, cardiac muscle cells, to do the job of cardiac output. New research, though, show there is some cell division, but how is it started? That's the question. And how could we, we talk about transplanting different tissue into the heart to see if it could rebuild some of the tissue, cardiac muscle tissue, post infarction. Infarction is death, necrosis of the tissue. Yeah. So of course, if you have dead tissue, it's going to impair the function of the heart, especially. Again, we're not doing heart, unfortunately, this semester, so we're just doing bone and muscle and nerve, basically. So you really have to know muscle. And we're doing only skeletal muscle. So this goes back to the developing tissues. Like this is even before mesenchyme. These are embryonic, first 8 weeks of Friday night, right? Friday night with the sperm cell meeting up with the oocyte. And these are our first tissue layers, ectoderm, mesoderm, and endoderm. We call them the primary germ layers. So when you're implanted, like Jeremiah, here's what Jeremiah looks like. And here's the uterus right here. And then you have different layers within that zygote. So much mitosis goes on. So you're going to have the 3 layers. Main layers are ectoderm, mesoderm, and endoderm. Like you probably know that mesoderm is probably going to arise the connective tissues. But the epithelial tissue is kind of spread apart. So I have to see what we're going to talk about. So it's embryonic development. And we did look at embryonic tissue or mesenchyme in lab. So nerve tissue, remember this because these are the ones we're going to learn. Nerve tissue arises from ectoderm. Nerve tissue. If you could remember that, that'd be really good. Muscle and CT arise from mesoderm. So the primary germ layers for those. So we're going to learn those. Epithelial tissue basically from all 3 germ layers. Technically, it's more ectoderm though. Okay, so this is just aging. What's going to happen? You're going to have less collagen, less elasticity. Tissue repair is going to take longer. This is a bummer. We got to reverse this though, right? We got to reverse all these things. And things start to get smaller, right? Bone loses its calcium, like through osteopenia. Hopefully not osteoporosis. Osteopenia means losing calcium from the bone. They're less dense. Muscle cells will get smaller. That's what atrophy is. Nerves start to degenerate a little bit. They get less jumpy, less excitable, if you will. And DNA changes a little more often. The cells are getting older. And there's things called telomeres. They're kind of like those ends of a chromosome that kind of degenerate every time a cell divides. The telomere, they get a little bit shorter and starts to cause senescence, which is it causes more risk of cancer and degeneration. The telomere is part of the chromosome that gets smaller. So what are we looking at here? The germ layers and where they grow this is how I started. I won't ever show you a picture of this, so don't worry. Okay, how are we doing? Are you guys still awake? So let's start the integument and mostly the parts. And I'll give you the physiology as we're going through what I think is important for you to know. Okay, so right away, the integument is a system, which means it has a group of organs, and organs consist of the 4 tissue types. So the integumentary system has all 4 tissue types, including the skin, which has all 4 tissue types, like the epithelial tissue is your epidermis. And then you have your CT, dense irregular connective tissue, which is your dermis mostly. So there's other connective tissue as well. Then you have the hypodermis, which are layers. One of the layers is mostly, I'm going to say, adipose, loose connective tissue, and areolar connective tissue. And then there's nerve tissue, because you need nerve tissue for sensory receptors, nerve tissue. And then you have the sweat glands and other glands, sebaceous glands, need motor neurons or motor nerves to contract or cause a release of those glandular secretions. And also there's a muscle tissue in there, and that's the erector pili. And that's, remember, this is smooth muscle. So you need nerve innervation to the muscle of the erector pili. All muscle needs a nerve impulse. The heart muscle, cardiac muscle, has its own ability to depolarize and cause a contraction. But it also is controlled by the nervous system. So I'm not going to talk a lot about hair and nails. Sweat glands we pretty much talked about, so we just got to label them. And same thing with the sebaceous glands, because these are mostly epithelial tissue, glandular tissue. So epithelial tissue is glandular, not only lining and covering, but also makes up your glands for secretions. Remember all those types. So here's the 3 layers of the skin. When I say layers, we're talking about the epidermis, the dermis, and the hypodermis. Hypodermis is also called the subcutaneous. Now remember, we did this in lab, so this is very, very similar. And we could also call that the sub-Q, hypodermis, below the dermis. So the epidermis has the strata, right? The 5 strata. And I always like to remember the bottom one is stratum basal, and the top one is stratum corneum. So below the corneum in thick skin is stratum lucidum. So that's thick only, thick skin only. And where's thick skin? That's on the palms of your hands, soles of your feet. Then you have the stratum granulosum. And then the one in between basal and granulosum is the stratum spinosum. And this is all, everything in here is stratified squamous, even though the basal cells look cuboidal, because you name them by the apical surface. So stratified squamous. And what's the protein? Keratin. So it's keratinized, stratified squamous epithelium, unlike the esophagus or the vagina. And it's avascular, just like all epithelial tissue. Dermis is vascular. Mostly fibrous connective tissue, which is dense, irregular connective tissue, not cartilage, connective tissue. The subq, also known as the hypodermis, and sometimes called the superficial fascia that connects things, is mostly adipose. shock absorbing, stores those triglycerides and insulation. Insulation is a good one when you think about where it is, below the skin, on top of organs, sometimes on top of muscles. So this is a good connection, like an anchoring of the dermis, which is the part of the skin, to other structures like muscles, right? Hypodermis. So there's always a layer of adipose, no matter how big somebody is or how much excess tissue they have. It's still part of the integument, part of the skin. Remember, adipose is loose connective tissue. It's connection to connective tissue proper. So this is the one you see all the time. This picture will be on the practical. So let's go to the hair follicle, right? The hair follicle. And that's all of this is hair follicle. Like if you said hair bulb or hair root, that's fine. Okay, it's fine. It's hair follicle. And then you differentiate the other parts, like this is sebaceous gland, which is attached to the hair follicle, and it's in the dermis. And remember, this is a sebaceous gland too. This is just cut, that's all. And then here's your erector pili muscle, which you can see is mostly in the dermis, connected here to this layer of dermis, to the epidermis, and then to the hair follicle. So always start with the layers, epidermis, and you can see how the bottom layer, the basal, kind of is wavy lines here, right? Papillary region of the dermis goes into the epidermis. So this is all dermis, and the slides are kind of difficult. These are little dermal papillae there. And then the dermis is the papillary layers on top. I know on the quiz that I'll probably give you tomorrow, if I haven't given it to you already, is, it might be confusing, but if you said dermis, you're okay. So the papillary layer of the dermis is here. Reticular is everything else above the hypodermis. So the second layer is the dermis. Then you have your hypodermis, or subcutaneous layer. So try to say, like, for the dermis, epidermis, and hypodermis. I remember epi means on top, hypo is below. Those are the layers of the skin. Right? And you can see the tissue. You can see the globules in the hypodermis, the adipose tissue. So again, here's, like, if you look really close, you may see corneum, lucidum, but you can't see the rest. The top of the skin, outside the skin, this part of the hair is called the shaft, the hair shaft. And there's pores throughout this skin, in between the, really what's from the dermal papilla, are the sweat pores. So most of the goodies are here in the dermis. Like here you could see a gland that's going up to the pore. So that would be a sweat gland, or eccrine sweat gland, if you like. That's fine. Just know the difference between a sweat gland and a sebaceous gland and visualizing it. And if you look at this other yellow tissue, like right here, these are free nerve endings. This is nerve tissue right here for pain and temperature in this case. These are lamellated or lamellar corpuscles for sensory reception as well, but these are more for pressure. Deeper in the dermis, right here is another corpuscle. Usually they're called Pacinian corpuscles. Pacinian corpuscles are nerve receptors. Sometimes there's some of those lamellar cells up here. I don't see them. They're called Merkel's discs, and they're also capsulated or lamellar nerve receptors. So know the difference between the nerve receptors, which is nerve tissue, the sweat glands, and the sebaceous glands. And then the red and blue, the bigger vessels, the blue is a vein and the red is an artery. So they're blood vessels. Here's another erector pili, and that's pretty good. It shows you it's being innervated by nerve tissue. So a muscle can't contract without that nerve impulse. So it needs the nerve connection. So all the tissue, all four tissues are represented. So the epidermis, these are layers, the 4 to 5 layers of epithelial tissue. And you have to know the strata of the epidermis. So it's a conglomerate. Skin is an organ. metabolism. So this is all collagen in here in the dense regular, right? You know what dense regular connective tissue looks like now. No cartilage in here. So that's pretty good. I think that basically does it all. So here's some terms you need to know. I'll get through this as much as I can. Keratinocytes produce keratin, which is the protein found in the skin. So of course, collagen is the most common and abundant protein in the body, and the most common protein, of course, in connective tissue. But in the skin, it's about keratin. It's a good protection, waxy. And these cells die as they get to the surface. There's so much mitosis, and in the end, the keratinocytes will fall off your skin, slough off. So they have connections. Your desmosomes are usually the way these cells in this tissue, epithelial tissue, are connected. You see this a lot. Millions slough off every day, and I showed you that last week. The dust bunnies, right, is your dead keratinocytes. Melanocytes produce a pigment, which is more protein, called melanin. So melanin is basically a brown pigment. There's two different types of melanin, but just know melanin is the pigment, the coloring, right? And it's more than just that. It's also protection. They protect the cell nucleuses from, say, the sun, right? Which skin protects our cells from the sun, but their cells make up the skin. So they need protection, and that's where melanin comes in. So the more melanin you have, the more protection you have from UV damage, because UV damage from the sun or any type of UV rays can actually change the DNA in your cells and cause things like cancer, especially skin cancer. And I'll show you the skin cancer. So I'm going to do exactly what I did on Thursday. Now, dendritic cells are Langerhans cells. So know this term, Langerhans, for the skin. It's, of course, in the pancreas as well, but these dendritic cells are macrophages patrolling the deep epidermis. So they're protective immune cells, activating the immune system, because they're going to give off chemicals too that increase vasodilation, bring in more blood and more cells of the immune system. So Merkel's cells, or discs, they're like Pacinian corpuscles that are sensory receptors. So those are the main players. That's pretty good. Here's the layers of the skin. Thin skin everywhere else besides the palms and the soles have 4 layers, and the thick skin has 5. So the one that makes the difference is the stratum lucidum. Now they kind of did it opposite of me. I put the corneum of the outer, I went from outer to inner, or superficial to deep. They're going deep to superficial. So any way you like it, but be careful with ordering, like on an exam. Like there'll be ordering on your lecture exam. Like I'll be talking about one of the questions on the lecture exam is like starting from subatomic particles going up to organism. Like name all those 8 or 9 steps, or those 8 or 9 levels of organization. So in this case, I have you order the right order from deep to superficial. So make sure you don't put it backwards, but I'll see if you did it backwards. I know what you meant. So you'll be ordering these layers, and I'll say thick or thin skin. So you just take out the lucidum if it's thin skin. So each one of them, again, for lecture now, again, same thing though as I did. The stratum basale is the bottom layer, and it has a basement membrane, and it's connected to the dermis, which is right below it. Highly mitotic. This is the most mitotic area. This is where the mitosis seems to happen the most. So of course, this is prone to cancer. So mitosis is nuclear cell division making 2 exactly the same cells. So you get one, you start with 1 cell, you get 2. And it's happening all the time. So those basal cells take, like a month to reach the surface, and they die off and become dust bunnies under your bed, the keratinocytes, which is still stratified squamous epithelium keratinized. So as you get higher into the stratum corneum, they die as they move towards the surface. So they die as they move more superficial. So the basal cell has no real fate except to become a dead cell quickly within a month. So other daughter cells, some cells stay in the stratum, like stratum. Well, this is stratum germinativum. Is another name for stratum basal. Germinativum is the same because they're germinating. They're going under mitosis very often. So not all of them are destined for the cell death. Some of them maintain the next level, which is the spinosum. Prickly layer. I don't get it, but when you look at the histology, you'll see it. We'll look at that tomorrow, and you'll look at more from home pictures that were given you. So it's a web-like pre-keratin. So there's starting to get keratin in there, but the keratin is more prevalent on the more superficial layers, like especially the stratum corneum. That's just like pig skin. It's very thick, dead looking compared to these other layers. Like even the stratum spinosum is very lively looking right above the stratum basal. So they start to produce keratin. So keratin's pretty much everywhere within the skin, but especially on the most superficial. And you have melanosomes, which are making the melanin or turning into your melanocytes. So cancer of those melanocytes is a really bad cancer, and it's got connections to the blood. Remember, the dendritic cells are your Langerhans cells, which are macrophages, immune cells. I think you got those down now. We mentioned that a lot. So now we're still going upward. Above the stratum spinosum is the stratum granulosum, because they're dark staining granular cells. Sometimes it's good if you have a lucidum, you can really see that starkly. So the appearance of the cells start to change. You don't see the organelles or anything disintegrating. You just see something flattening, becoming more squamous looking, and you got heavy keratinization there. So the granules are basically making keratin within the stratum granulosum. And keratohyaline, so that implies that cartilage type granules forming the keratin fibers in the upper layers. So now from the granulosum up, obviously the next layer is corneum without the lucidum, has high amounts of keratin. And you also have these other lamellar granules. So mixed in with the keratinocytes is this glycolipid. So it's a little carbohydrate lipid. So lipids are hydrophobic, so it kind of has a waxy type of consistency to it, especially when you look at it under high magnification. Kind of looks like strings of wax. I just thought of that, you know. And then everything above that is dying into the stratum corneum, which will be sloughed off. So you can't get blood supply to that level. It's impossible to diffuse right through all those layers of stratified epithelium. So it's hard to get the nutrients up there. So they're going to die off. They have no chance. That's some really rare disease where you do have blood supply there for some reason. I don't know of any. Stratum lucidum is translucent, which means it's kind of transparent. So these are still dead cells that kind of look empty, and that's why, because they're dead. Keratin, the protein, lives on a little bit, you know, in time until it becomes a dust bunny under your drawer. And its lucidum is only in thick skin, and it lies superficial to the stratum granulosum. So it's between the stratum corneum and the stratum granulosum. Stratum. So stratum corneum is the horny layer because it's like thick and kind of like pigskin layer. And many rows, that's a lot of rows of cells when you think about it. So it's kind of dead looking. You know, you can't see a clearly standing nuclei, let alone a basement membrane at the bottom, because it's not there. And keratinized dead cells. So void cytoplasms. So that's a lot, 3/4 of the epidermis. Dead cells, but they act as protection. They have reason. This anatomy is definitely helping with physiology. It prevents water loss and dehydration. And this is the main thing you're going to say when you look at the histology of any stratified squamous epithelial tissue that's for protection from friction or abrasion, and a barrier for penetration, and also a barrier for microbes like dangerous bacteria, fungus, parasites, virus, and other chemicals, and other physical assaults too, as much as it can be, right? So apoptosis is a controlled cell death. It's not necrosis. It's when a cell has lived its life and now it's been programmed through its cell cycle to die, and that's completely appropriate. So the cells slough off, especially with the stratum corneum, dandruff, dry, dead cells, dander from animals, mammals, and us, I guess. So we shed 50,000 cells every minute. I can't even count how many we've been here so long. Hope they're getting replaced. So here's kind of a cartoonish version of this whole thing, although the left side looks like a really dark staining, real histology here. So look how thick the stratum corneum is. It's just layers of wiry, waxy looking cells. All this is stratum corneum. Remember, it's like 2/3. Is that what it said? 2/3. That's a lot when you think about it. Or most of it anyway. Don't worry about the 2/3. And then there's no stratum lucidum here. So this is thin skin or anything but thick skin, and you can see the granulosum. It's pretty good here. You can see the darkening of the cells. It's subtle, but it's there if you really look, and it's consistently through that layer. High power. And then below that is a lighter staining stratum spinosum. You can see the difference in the cells here too, like the spinosum. And basal technically is like one layer, right above the dermis. All of this in here, below this. So see where they separate. Spinosum, granulosum. I know it doesn't help when I write over this, but at least it gives you the idea. And this is all dermis down here. Invaginating up. That's where a lot of students get lost when they see this. They call it stratum basal. Takes a lot. You have to look at this a lot. So the dermis, again, this is where all the goodies are. The fibroblasts and the immune cells like macrophages, mast cells that secrete your histamine, your neutrophils and lymphocytes, which are white blood cells. And the matrix are fibers. They could be collagen, of course, reticular fibers and elastic fibers. But remember, the dermis is dense, irregular. So it's not as dense as the dense regular connective tissue, like a ligament or a tendon. And the upper layer is papillary. That's a small layer just below the stratum basal of the epidermis. So layers and stratum drives you crazy. And the rest of it is a reticular layer. Connective tissue, dense. This is a really good picture. This one really has a lot to offer here. And you could see, like, this is all, anything below this red line is dermis, right? I should have wrote that here. So this is all dermis here, right? So this one layer is your stratum basal. One layer just above the dermis. And then you have stratum spinosum, is that lighter staining stratum. And then you have the stratum granulosum right in here. Dark staining. So technically, I don't see it. These are just separations of tissue. So all this is dead-looking corneum. This is all stratum corneum. And this right here, you really can see the dense, irregular look. It looks just like the slide we saw of the dense, irregular connective tissue. A lot of review. I know it's a lot. So a superficial layer of the areolar connective tissue. And it's basically connecting the dermal papillary to the epidermis. That's really what, and it's areolar. So there's another layer of connection of tissue. So it's connective tissue. So within that dermis, the top dermis part, the papillae, you have those sensory receptors. like tactile receptors, touch receptors. Meissner's corpuscles is another one. There's another corpuscle you have to learn. The Pacinian were deep pressure. The free nerve endings are there as well. So there's pain receptors. It's all nerve tissue within the bottom of the epidermis and the top of the dermis. That's where you have a lot of those receptors for pain, temperature, fine touch. And that's what those receptors are for, those tactile. Tactile means touch. Tactile receptors. Okay. In thick skin, that's where we have our fingerprints, right? And then you have these dermal or epidermal ridges. And that's all you really have to know, that the dermis, dermal, and epidermal ridge give you the friction ridges, which leave that fingerprint pattern. We don't have to go through fingerprints. We'd spend the whole day talking about the skin. We don't need to, because we have to figure out what tissue is and move on to a tissue like bone soon, next time. Okay, so the reticulare is the big part of the dermis. Dermis, the layer, coarse, dense. There's also elastic and reticular fibers. Keeping the skin hydrated, you can tell that's going to happen. This is actually the dermis, and the reticular part of the dermis is actually what we use to make leather. Right? So when you hear the term plexus, that's the network of blood vessels. It could be nerve too. So the hypodermis is below the reticular layer of the dermis. And that has more pockets of adipose cells. And you could have pockets of adipose cells in the dermis as well. But that's mostly dense, irregular connective tissue. Cleavage lines or tension lines in the dermis caused by the way the direction of collagen fibers. Like in your neck, you have like tension lines. Like in, God forbid, you had to have like neck surgery. They may like for a thyroid removal or something like that, or to go in the front to do something, they try to cut along those tension lines so you don't get a scar. And that's what they're talking about, these cleavage lines. Sometimes they call them age lines. I don't know, ask Kylie Jenner. She'll tell you how to fix those. But plastic surgeons are very careful so they don't leave a scar, and they try to cut around into those tension lines because they're already seen, so they would look normal. You don't have to know this. This is a little spooky to me, but it's showing all the cleavage lines that could happen based on all the directions of collagen below the skin in the dermis. Really, the epidermis is just dying off, but the dermis is what gives the skin the shape when you think about it. That's probably why we wear coats and wear shoes made of leather. And they're definitely waterproof in most cases. They prevent water from getting too close to your feet anyway for your shoes. So flexure lines now are reticular layers and dermal folds near joints. Not important. More dermal markings. I want to go down to burns, stretch marks. I'm not going to ask you about stretch marks. Skin color, we mentioned pigment, and melanin, the eumelanin, which is the brown melanin. So there's more melanin you have, the darker your skin will be. The less melanin you have, the lighter your skin will be in color. So that's really all you need to know about that. Damaging, of course, UV light destroys the skin. You don't have to know about the folic acid, but it changes the DNA synthesis, which can cause cancer. Carotene is the yellowish pigment, and vitamin A is based on carotene. This is important for your retina rods and your eye, your photoreceptors. That's why I say eat vitamin A. It's good for your eyes. So that's good to know. And that's a yellow-orange pigment, like in a carrot or an orange. And that accumulates in the outer part of the skin. So vitamin A, carotene, I think it's important. Hemoglobin. Yeah, this semester we're not talking a lot about hemoglobin, but it can change the hue, so to speak, hue of your skin. I'm not going to ask you that. I like this. I did this on Thursday. Like cyanosis means blue. Pallor is like blanched or pale. Erythra means red, like a red blood cell is erythrocyte. So it's good to go through these. And the cause is hypertension and inflammation, right? Vasodilation, bringing in other cells. So when you have tissue damage, you get erythema, which is inflammation, vasodilation, more blood to the area. Jaundice, because of a problem with the liver, hepatitis, cirrhosis, other liver destructions, liver cancer, can cause your skin to turn yellow. So we always examine somebody, look at their skin first to see what they look like, if it gives you any clues, right? Bruising, don't care. I mean, I care, but we're not going to talk about bruising and how that happens right now. We'll just learn tissue repair, right? Hair, we're not going to really talk about. Just know the hair follicle, know all the structures in that area. Nails, I'm definitely not going to talk about. I think the only thing is the ABCDE rule and the burns. So I'm going to keep this pretty much lecture and lab pretty much the same. Cerumen is earwax. The ceruminous glands, you should know that. Like sebaceous gland of the skin secretes sebum. Don't worry about holocrine or anything like that. Just know the names. Get a good outline of this. And you might see histology like this. Again, I told you this on Thursday. Like, this is a sebaceous gland here. And then you have your hair follicle, which basically has stratified squamous epithelium building the dead cells into the hair, which is dead keratinized cells as well. Hair today, gone tomorrow. Sweat glands are basically just know what they look like, as opposed to a sebaceous gland. And this is also a sweat gland. So eccrine is the most common one. And then we have apocrine, and they look very similar, right? Apocrine is a little just heavier sweat. And then sebaceous glands are much more viscous, heavier sweat, the heaviest. Not sweat, it's not a sweat gland, it's a sebaceous gland that secretes sebum, which could have some antibacterial effect, and it's oily, waterproofing, more so. Functions of the skin, protection, these are important. Body temperature, this is a question you get. Controls, keeps the heat into the body or the cold from getting in, so to speak. Sensations is nerve tissue. Metabolic functions, converting vitamin D. This is important. So you do need the UV radiation to convert vitamin D. Make vitamin D. And you need vitamin D to absorb your dietary calcium. So this happens in the epidermis. Blood reservoir, especially in the dermis and hypodermis, because there's a lot of blood vessels in there. So the blood sits there waiting to be used for something. That's what that means. It's hard to understand that without understanding the cardiovascular system. And you sweat out waste. You sweat out carbon dioxide, actually, in very small amounts. Protein breakdowns, but of course the kidneys do more of that work. So those are what you should understand about this chapter. And it forms a barrier, a chemical barrier, of course, a physical barrier, that organ that we call skin, and a biological barrier. So how does it do that? Skin secretes chemicals, antimicrobial, like antibacterial, killing bacteria. Sebum is part of that. Defensins or other proteins, which are antimicrobial. These are fairly newly seen. And the skin basically has a low pH, so it's kind of an acid mantle, not like Mickey Mantle, we're close. And it's for preventing growth of bacteria. So it's an acid mantle. That's pretty important. And then we know about melanin, the chemical that stops the danger of UV radiation. It doesn't inhibit the production of vitamin D, though. Physical barriers make sense. Remember the stratum corneum and all those waxy parts of the skin, keratinocytes, blocks, things that can come in like water, right? And limited entry of lipid-soluble substances and these allergens that are from the poison ivy plants, which if you're allergic to it, you're sensitive to it, of course, you're going to get a problem, an inflammatory response, immune response. And of course, it depends on the concentration of these things, like acetone is in nail polish remover pretty much. So it has a very strong chemical to it, but it depends on the concentration. The lead and mercury, of course, can't get into your skin. Hopefully you don't swallow or eat any of that stuff. So a limited penetration of the skin. Nitroglycerin is pretty lipid soluble, but still. So again, it's very impermeable is the idea. And I'm not going to ask any specific questions, but understand the protective part and what it protects against. So biological barriers, we have the macrophages, the Langerhans cells, dendritic cells, engulf, right? That's what a macrophage does, engulfs. Right, so that's what this is all saying. So, of course, the melanin. Sensory receptors. Extero means outer, and you can respond to certain stimuli. There's our picture. You should really know this well. You should really, definitely go through this well, know all those parts that we labeled. And tomorrow we're going to do it again in lab. All right, you have to do that again with maybe a model. So let's see if you take pictures. So skin can synthesize vitamin D, because without vitamin D, you can't absorb your dietary calcium in the small intestine. Yeah. And keratinocytes may have some help in stopping the mitosis of cancer, because they undergo apoptosis. So there might be some factors that could slow down the mitosis of other cells. And activate hormones, convert cortisone into hydrocortisone to make it more active. So there's a lot of functions here. So here's the last thing we'll do, the skin cancer and maybe some of the burns. Remember the ABCD rule? Is it this asymmetrical? What's the border like? The color, the diameter, the size, and is it elevated? You can't just have one and call it cancer. They have to be a group of at least 3 of these each. And the 3 burns, we're going to stick to first and second and third degree burn based on the layers of the skin. So basal cell carcinoma is the least malignant. It is malignant, but the least dangerous. Squamous cells in between. And then melanoma is bad, the worst. Most malignant, most ability to metastasize and get into the blood and go to other places. Metastasis means it goes to a different tissue or different organ. So basal cell carcinoma, let's take a look at it. I'll put a picture on the exams. So basal cell carcinoma can move its way up and invade downward too into the dermis, because now you know where the basal cells are. And this one is usually what's called in situ, which means it doesn't spread. It stays in one place. So it's kind of good color, not too dark. It's symmetrical. It's not huge. And it's only slightly elevated, and it looks pretty regular around the margins. Basal cell carcinoma. Squamous cell involves, of course, keratinocytes and other squamous cells, as we're getting closer to the superficial. And this is a scaly, I've probably seen people with these things, like scaly papilla, little bumps on their ears. Yeah, I've seen this in lower lip, and it just doesn't go away. So, of course, you got to get screened by a dermatologist and maybe do a biopsy. A biopsy is actually removing the tissue and looking at it under a microscope and doing all types of tests to see if it's mitotic tissue and cancer nuclei, damaged cells. This is the squamous cell carcinoma. So now we're getting kind of a big diameter. It's kind of weird elevations and it's a little asymmetrical. The color is mottled. And the time really should be added to that equation. Like, how long has it been there? Did it change? And here's the ABCD rule: symmetry, the border, the color, the diameter, and E would be, is it elevated? Flat. Sometimes it goes downward. It's such a destructive cancer, even squamous cell. It's destroying the dermis or basal cell too. So again, this looks really dangerous, and it is. It doesn't. I mean, you still have to do a biopsy. You don't know. I mean, you would never just look at something and say that's melanoma. You'd have to take that piece of that tissue out deep too, because this would, if you open this up, you can see it's going deep into this. Remember, this is elastic cartilage, right? Elastic cartilage around here. So melanoma, dark color, asymmetrical, irregular margins, border diameter could be really weird. Like the basal cell, like I said, looks kind of like Bermuda, but the melanoma could look more like Hispaniola, different angles and colors and separations. So the burns, I'm not going to do the rule of nines. So here's what you have to know of the burns. First degree only burns through the epidermis, stops at the dermis. Second degree is epidermis and dermis. Third degree, let's see what they say if there is one. Third degree is all the way down into the hypodermis. So believe it or not, this is the least painful, but of course the most dangerous, because all the nerve sensory receptors are up here. So if you kind of still have these intact, you're going to be in severe pain quickly. But if you get into these, at first you're not going to be in pain because you're numb. But of course, when this comes back and those nerves come back, this is terrible situation here. And you're going to get dehydrated because your blood vessels are exposed, losing fluids. So that's it. I think we hit all the important parts of the integument that you need for your